Talks in a few minutes here, so you want to just grab a refill if you need one right now, and grab a seat if you need one, and we'll get going in a sec. Was that your video, or that was like from?
That was a friend's video.
Yeah.
Somebody.
Yeah. Amazing. All right. All right. You're gonna be able to connect, or?
Yeah, I have my own.
So Stefan was expecting some questions, so I'm gonna keep talking a little bit so we don't get distracted here. So Stefan Doerr, from Acellera. So Stefan's going to be talking about some of the new stuff that's happening that we're seeing in the world as a whole. More AI related especially to dynamics, and I think that's a very interesting area where we've seen a lot of promising results coming out recently in publications. So Stefan's going to talk about ACEMD-ai, Ab-initio machine learning calculations of small molecule quantum energies and forces. Take it away, Stefan.
Thank you. So, yes, I would like to thank the organizers also for giving me the opportunity to present this work. I would have to disappoint you, I don't have any cool videos of caves or any other such interesting stuff, but I hope you'll find it interesting anyway. So, I'm going to talk about ACEMD-ai, or ACEMD Ab-initio as we call it, and specifically about the calculation of small molecule quantum energies and forces. So this is a vision forward, not specifically our vision forward, because we didn't start it, but we definitely believe in it, we subscribe to it. So as we all know, classical molecular simulations are very useful. The speed and the system size is increasing steadily with the advances of GPUs, but we are limited by the accuracy of force fields. On the other hand, we have quantum molecular simulations, which give us many advantages. However, they are too computationally expensive. So the idea forward, in our opinion, is are the QMML networks. QMML are machine learning methods, practically neural networks, which try to learn the energies and forces from QM calculations. We're trying to build essentially a many-body neural network potential. The idea is that neural networks are in theory able to approximate many kinds of functions under very mild assumptions, so we believe that we can approximate the energy potential from QM, and they also allow us to calculate very easily the gradients of the networks, and this will come in handy for calculating the forces. They have been around in general since the 90s. They've been used mostly in material science, but recently, because of the advances of deep learning and GPU acceleration, they've become again quite popular. So there have been some successes recently by Jörg Bello, the team behind DANI1, the team behind TensorMall, and DeepMD. We might be missing some, but these are the ones that I know of. So to give you an example of how QMML networks work, essentially you need to convert your atomic environment into something that a neural network can use. So we are trying to describe the environment around an atom, like this nitrogen here. We are trying to describe it, in our case, the featurization we use is trying to describe it through some radial information, like the distances between the atoms, and also angular information about the angles. And this information is practically represented as a vector and is fed inside the network. So it's kind of a small vector of 300 elements. So we propagate it through our network and we output the atomic energy. So then what we do, for example, for a molecule, we take our element network. So we have a network for hydrogen and a network for oxygen. So we apply these element networks on the description of the environment of each atom, and we output the atomic energies and we sum them up to the total energy. So then we can train these networks to minimize the error both in the energies and in the forces. And this is the loss function that we use, which we have taken from, I think, from a DeepMD method, which penalizes the deviation of the energy of the network from the DFT energy, and also of the forces of the network to the DFT forces. So we did some preliminary, so these are a bit preliminary results that I'm going to show you. So we simulated a data set using with DFT using this functional and base set. Our training set consists of around 3 million normal mode sampling conformations. It is based on the data set by Yani1. We subselected it to kind of the lower energy conformations for the moment because we didn't have time to simulate everything yet. So there are up to 8 atoms in that data set. And during training, we are able to reach practically, we're able to predict the energies with an accuracy of 0.6 kcal/mol, and also the forces with a quite good accuracy. Our validation set comes from the same database, GDB11, but it's actually larger molecules containing 10 atoms, all of them. And we have there 1,126 NMS conformations, and as you can see, so here we call it GDB10 because it's, or actually the anyone people call it GDB10 because it's the GDB11, but only up to 10 atoms, only 10 atoms actually. And as we can see, the energies, the predicted energies and the actual DFT energies correlate very well. Right, so the biggest advantage, obviously, of this method is that if we look here at the runtimes of doing a QM simulation, for example, compare it to the time that we need to do an MD simulation, there is a huge gap between them. Obviously, MD scales linearly with the number of atoms, QM doesn't. So our method, ACEMD-ai, or QMML networks in general, they scale linearly as well. In this example, it doesn't scale linearly because it hasn't reached yet the linear scaling, but if we were able to simulate larger systems, we would see that as well. Still, as we can see, there is quite a gap between the performance between MD and QMML networks. So I don't believe that will be a substitute yet for normal MD simulations. But I'm going to present to you one application which we have been working with, which is on small molecule parameterization. So, in our lab we like to do MD a lot, but as you know, charm and number, they don't contain parameters for small molecules. These parameters, if we want to obtain them very quickly, we could use GAF2 or CGNFF, which use some empirical rules to apply pre-calculated parameters to the new molecules, which we are about to simulate. However, these are very often not very accurate. So we have developed this Play Molecule parameterize application. So Play Molecule is a website. You can visit it on www.playmolecule.org. One of the applications that we have there will be for parameterization. And currently, it does QM simulations, so it improves the charges of the molecule by fitting to ESP calculations. It improves the hydral parameters of the molecule by fitting to the QM energy potential. And however, the problem with this, as I was saying, is that these are QM calculations, so they are very slow to perform. So what we want to do is practically to substitute DFT with ACEMD-ai. So the pipeline that we use for parameterization is the following. We get the initial parameters from GAF2 or CGNFF. We minimize our molecule using ACEMD-ai. We get the gas tiger charges also from GAF2. We detect the rotatable dihedrals on the molecules. We evaluate the energy. So we do a dihedral scan practically. We evaluate the energy using ACEMD-ai, and then we try to fit the parameters of that molecule to the dihedral energies. So to give you an example, these are some simple molecules that we are testing on. Yellow is the DFT energy, and blue is the ACEMD-ai energy that we're predicting. As you can see, we're still not there. So, for example, we don't get very well the peaks because we have trained on mostly low energy data. However, you can see that the minima are in the correct positions, and also the maxima, which is quite important actually. So, if we take these energy calculations that we have for the dihedral scans, and we try to fit the initial parameters from GAF2, It looks kind of like this. So red is our energy calculations. We start from the green parameters, energies from the parameters, and then we fit it, and we are able to fit them very well to the ACEMD-ai energies. So as I was saying, one of the biggest advantages is practically the speed. For the moment, we did some calculations for 40 compounds from PDB bind, and we found out that to parameterize this with QM, it would take us 474 days, CPU days. With GAF2, it's very quick, so it takes us around 6 minutes. With ACEMD-ai, for the moment, it took us around 4 hours, but this is practically the upper limit. So we haven't tried to optimize our code at all. Well, a bit, but not much. So there is lots of space there for speeding up our method, and we think we can reach performance close to GAF2 eventually. So in this sense, I think that it's a very useful tool for getting better parameters for small molecules. And yes, so about the availability, as I said, the parameterized application is available on PlayMolecule, playmolecule.org, so for free use, or Acellera also provides private installations. ACEMD-ai, the code, it's written mostly in PyTorch and Numbacuda, and it will become available at publication. But if you want to collaborate with us before that, you can request free access to our Git repository through this mail, developers@acellera.com. So to conclude, we have these QMML potentials, which are trained on energies and forces. They can scale linearly with a number of atoms, and they are much faster than the QM calculations. And we are trying to apply them practically for small molecule dihedral parameterization. There are still a few challenges, important ones. So we can improve the speed. We hope to improve the speed of this method. And we also need more data because we need to train it on QM data. We need to do lots of DFT calculations. So both for molecules, so conformations with higher energy, also some charged molecules or larger molecules in general. And if we want to do simulations, we also need to treat somehow the long-range electrostatics. So I would like to thank my colleagues, especially Dominic, who did lots of work in our group, Raimondas, who also works at Acellera and does most of the QM stuff, and Gianni. And if you want to talk with us here at the conference, you can talk either with Gianni, me, or Frank, or visit us at the poster session on Tuesday. So that's for the talk. If you have any questions, feel free.
Great, thank you, and great with the movement to collaborate and open it up to everybody. That's fantastic. Questions? Yes?
So can you say something about the ability to extrapolate versus just interpolate the space in which you've sampled as far as forces, et cetera, goes?
Yes, extrapolation is probably tricky, but what is an advantage is that you can detect when networks are extrapolating. So there are some methods at least. So you could have multiple networks and see where their predictions vary a lot. So they will probably be trying to extrapolate in those regions. For the moment, well, some of the examples actually, the GDB10, for example, it is an extrapolation because we've never seen that many atoms inside an environment. So by itself it is, but if we want to extrapolate to much more cluttered atomic environments, I don't expect that would work yet. That's why I said we need more data, larger molecules. So, well, okay, that will be interpolation at that point, but yeah, I think being able to detect when you're extrapolating is very important for the networks. Any other questions?
When you're learning your networks, can you ever learn about the underlying physics from how the variables are trained?
I don't know about that. So I cannot. I'm not Sure. So in which maybe I missed somehow the question, but not.
Do you learn anything from the?
Personally, if we can learn from the network. Okay. I see. Well, neural networks, sometimes you can look at how they operate. Like with convolutional neural networks, it's nice to look sometimes at the filters that they produce for local regions. In that sense, you can see what they are seeing and how they are interpreting the image. In fully connected networks, I'm not so sure. So I cannot tell if we could learn something from that, but it would actually be very interesting.
Yeah, excellent question. Yeah. Great. Well, thank you, and great presentation, and plenty more time to sort through that. And I think in the AI space, that's probably one of the biggest things is getting to what's really happening and whether we can actually learn something from this. They seem to be very predictive once there's the right data out there, but when do you know if there's enough data to make a good prediction, and what can we actually gain from that? So, all right, let me just get everything set up here for our last presenter. All right, so it was mentioned that there were going to be 4 presenters. There are 3, so everybody gets one for free. So our last presenter is Sergio Decherchi? Decherchi. From BiKi Technologies. So he's going to be talking about kinetics of protein-ligand unbinding via smoothened potential molecular dynamics simulations. Take it away, Sergio.
Thank you. The surname is difficult also in Italian. So okay. I would like to really thank the organizer for this possibility, and today I would like to introduce you a very simple and quite effective method to rank ligands, rank drugs based on their residence time. Why K-off? Well, K-off is a pretty important parameter. The residence time is considered now a pretty important parameter to be studied together with classical thermodynamics. In particular, one of the reference papers on this field is from Copeland, which observed, it was not the only one obviously, but it was able to observe that clinical efficacy was significantly well correlated with the K-off, the residence time. In some cases, maybe particularly for enzymes, much more than thermodynamics. Together with other nice properties of nice features of having a good residence time, such as drug dosage and reducing the potential side effects. So K-off is a key parameter in drug discovery. This is something that is assessed today. The first approach could be doing experiments. If you go to a classical, let's say MedChem, the first say will tell you, okay, let's do an experiment, let's do an SPR. Still, doing SPR experiments is not so trivial. There are some problems that could emerge. For instance, first I have to express my protein. In some cases it's easy, in some cases it's not that easy. You have to immobilize your protein in the surface when you do this kind of experiments. You have the dependency of results on pH, and you have the possibility to have an inactive target. So you have many variables that you have to control to run SPR properly and estimate K-off. So it's not trivial to run this kind of experiments. So that means that, actually this means that we have an opportunity as computational scientists, that is that we can try to use some computational tools to try to address the problem, to try to prioritize our drugs based on the K-off using, for instance, molecular dynamics. If we do classical, plain vanilla molecular dynamics, clearly that could be completely useless, because it may be you need the age of the universe to simulate by plain molecular dynamics the unbinding process of a drug, of a nanomolar drug. So okay, plain molecular dynamics is not the answer for this kind of problem. So we need some kind of smart methods. Several of them are well represented in this conference, such as Markov state modeling, several free energy-based methods such as metadynamics, transition path sampling, milestoning, metadynamics, and the last, that what we introduced and proposed as a solution is ScaleMD. Well, this is a possibility to move from free energy to kinetics. This is, let's say, a vanilla approach in which you define a reaction coordinate of your unbinding process, and this is difficult. This is usually difficult, and to some extent tautological in perspective scenarios. You compute the free energy to an high precision level, whatever it means, high precision level, and then you get your K-off using an Arrhenius-like equation. That could be done, but you have several problems in doing something like this. And the slide is done exactly to show you that, try to convince you this is a possibility, but this is a difficult possibility to be done, to be performed in practice, because this is a very ill-posed, mathematically speaking, problem. Because in order to have a reasonable estimation that the K-off, you need a very high precision on the delta G of the exiting of the ligand. So this is very difficult practically. You can do that, but probably is not the simplest way you could do this kind of, the simplest way you could approach this problem. So what we propose is very simple conceptually, and it consists in smoothing the potential function, starting from the original one, lambda equals 1, where lambda is a multiplier of the potential function, and then by decreasing this lambda value, you can flatten the potential and increase the probability of events. This is not different from the Boltzmann viewpoint of increasing temperature, still numerically is not exactly the same. So our strategy is very simple. Decrease lambda to a certain level that is problem dependent, accelerate events to get your unbinding events, and average out in several parallel replica your results in terms of average unbinding time. So the protocol is absolutely simple, but it has the advantage of being trivially parallel, because if you have a cluster, you can run all those simulations in parallel and then get your unbinding times. Clearly, you cannot do that alone. You cannot just scale the potential and expect not to fault the system in some cases. So here, the real free parameters are the scaling factor, the lambda value, and restraints. By restraint, I mean a set of restraints to be put on the backbone of your protein, except the nearby of the ligand, that is the binding site. In this way, we are able to accelerate locally in the nearby of the ligand and without compromising the folding of the protein. Optimal values of these scaling factors and restraints, in some cases, are very easy to be obtained. They need a bit of calibration. In some cases, are more difficult. But a huge advantage of this method is that you're not defining any collective variable. You're not defining something a priori in which you expect, for instance, your unbinding path or something like that. So it is completely reaction coordinate free. And this is a huge advantage in perspective studies when you have to apply it to real world pipeline, industrial pipeline when you develop a drug. So we have these n-independent unbinding events. We get an average unbinding time. And now, if we consider, again, the Arrhenius relation with the scale potential, what we get is something like this, the enthalpy and entropy in terms, where we don't know exactly what happens on this part here. However, assuming that in our congeneric series, because we are assuming that, we have that this term here is reasonably similar across the ligand, then it's from simple algebra, this can be written. That means that the ratio of the simulated scalar times is linearly is linearly correlated with respect to the physical unbinding time. This is a simple trick to try to linearize the relation between the simulated time and the physical one. Another thing you could do, this is very easy, this is just simple computation, given the mean, your mean time, your standard error, then it's easy to run a Monte Carlo simulation to assess which is the probability of a ranking. That is, I have called ranking B, ranking D, ranking F. For instance, L1 ligand 1 is lower than ligand 2 is lower than ligand 3, and all of them represent, all of these letters represent one specific ranking. It's easy to assess which is the probability of that ranking given the means and the standard errors. This gives you some help in understanding how many simulations you need to try to say, okay, this is the final ranking from my data. So we applied this method in a first publication to the adenosinic receptor, then to the heat shock protein, and then to GRP78. In later studies, we applied, in collaboration with Servier, to glucokinase, another complex from Servier that I cannot say you because we signed an NDA, and very recently to stem from fragments from Aztecs. As you can see, we cannot expect a perfectly linear behavior because we have not control in some of the terms that I show you. Still, in terms of ranking, the method proved to work quite well. Like in this one, here you have the physical rates scale with that formula, and here the simulated times. Same thing, something similar holds for this system. Again, is not perfectly linear, but still the ranking is pretty reasonably obtained. That is what MedChem wants to study the ligands and try to modify them to increase the residence time. Same thing holds for these ligands that were from a series from hectares. Even in this case, the ranking was pretty good. Then we tried to challenge the method a bit more. That is, here in this series from Servier on glucokinase, the conserved part, there is no scaffold actually in this series, no conserved part. So here in this way we tried to push the method even more, because here the entropic terms could play a major role with respect to the other ligands. And what we found still, is that more or less, in this case these two were two enantiomers, and we average out the results. Still, the ranking is quite preserved, except for this ligand here, that quite interestingly, guys from Servier said us that probably they were not completely sure about if the experiment was completely right in that. So that was pretty interesting. And we published the results recently on Medicina Chemistry Journal. Another case, pretty interesting, is from Aztecs. They gave us a series of compounds, of fragments that were bound to a very shallow pocket of this XIAP protein that is involved in cancer. This was studied only experimentally from Aztecs. And now what we wanted to test is the ability of the method to deal with fragments, in particular fragments bound to this superficial pocket. These fragments were mimicking a protein. And we got two series of results. The first one is the ability to split in millimolar, micromolar, and low micromolar fragments, and then changing the lambda, changing the scaling, we were able also to find also in this graph the nanomolar inhibitors. So also in this case we got a pretty good result. The last example which we applied, those slides are from Servier. I would like to thank Pierre Ducrot for allowing me to show them. I cannot say you about the system, but it is a protein-protein complex. And this study was completely performed independently in Servier. And in this case, the number of compounds was 71. So the chemical space was pretty good, and still a good correlation was achieved. One thing that I didn't say before is that in this case, if you consider free energy perturbation, considering a fragment that is on the surface of a protein, so not a real pocket, a very shallow pocket, with free energy perturbation method, we will have a lot of problems in this case. Because when you are morphing your potential, what happens is that the ligand most probably will fly to the solvent. So given this, you probably won't get any convergence with that kind of method. Sorry. The very last thing I would like to say is that in the ScaleMD methodology, we are doing something very rude somehow, because we are injecting noise into the system in a completely agnostic way. What we are working now is a gentler method in which we are more oriented to the unbinding process, in which we still use a reaction coordinate, but we can somehow give it for granted, a default, if you want, in software for the reaction coordinate. I just tell you that this is based on the so-called adiabatic bias molecular dynamics, and the great advantage of this is that you don't need any restraint of the system, because the kind of acceleration you have is very focused on a specific part of your, that is the ligand and the binding site. So a brief summary of the results. What we verified is that in a congeneric series ScaleMD actually works practically pretty well. The amount of time that you need to run this kind of computations is pretty reasonable, because it's trivially parallel. So even if you have, let's say, 20, 35, 50 nanoseconds to get your binding, then you can run all the replicas in parallel, so it scales very well if you have a cluster. Most of the time you get a correct or almost correct ranking of your ligands based on the K-off. It can be applied in cases in which, for instance, free energy perturbation can fail because of the morphing problem on shallow pockets. In that case, you correlate with IC50 or KD, but you can assume you can correlate with that if you assume a diffusion process, a diffusive process for the K-on, or the K-on is similar for all the ligands. And also, something that really didn't do till now, but what you can do is to build a regressive model, given your data, to try to extrapolate the absolute K-off value. I don't have any clue, honestly, on how much is reliable this, because we never did it, but actually it's something that you can do. An important aspect of all these methodologies is that you need a good co-crystal structure. If you have a docking, if you have a starting structure that you don't trust that much, okay, forget it, it won't work. But if you have a good co-crystal structure or small substituents such that your pose is reasonably that, then we have pretty good results. Thank you for your attention.
Great, thank you, Sergio. Questions? Yes.
Any idea how this works for on rates?
In the case of on rates, well, the point here is that you are injecting noise to the system, and you want to move from a co-crystal structure to the ligand-free into the solvent. So this is a destructive process. What you're asking is a constructive process, but injecting noise to get a constructive process probably is not a good idea. So I guess this is definitely not the right method to get a constructive process. What I can tell you is that we developed another method not to estimate the K-on, but to accelerate the binding process. We have the symmetric part of this that is called MD binding that we recently published, but is completely different, a completely different approach.
How do you treat the cases where there's a, say, a gatekeeper of the binding site, some conformational change that opens up the binding site, allow the ligand to dissociate?
What we usually do to try to guess which are the main elements that has to move in your unbinding process, what we do is to compute the pockets geometrically. We have a tool to do that, and then based on those pockets we release, we set the restraints on all the backbone of the system except the residues involved in facing the pockets. So it really depends on the fact that those, if you have a huge channel, okay, and you are not able to identify well this pocket, then you need some a priori knowledge in your system. But as long as your residues are in the nearby of the ligand in a reasonable way, you can do a pocket computation, see which are the residues in the nearby of the ligand and release them.
Hi Sergio, nice talk. So just try to be a little bit provocative. You say you can get rid of the reaction coordinate, but at the very end you need to know which are the slow mode of your system, no? At the very end.
Well, when you set the restraints, actually you need to have an idea of what is going on, clearly, so you don't get rid at all of, but still, my personal guess on this, and what we got also from customers or people that is using it is that this kind of reaction coordinate is not tautological, is relatively easy to set up it practically. So I'm not saying that you don't have to need anything about the system. You still need to know something, but still you don't need 5, 4, 6 reaction coordinates to accelerate your unbinding process. So it's a weak version of a reaction coordinate, if you will. Still, but I agree with you that conceptually, still you need to know something. But the probability that you don't know anything about your system and you're studying it is pretty low.
Last question here. Sorry, we got some. I think things are getting rowdy in the back there. So.
Thank you very much. Very interesting talk. I'm sorry if I missed it, but how do you tune the scaling or smoothing, as you mentioned, and also how many simulations are required per ligand in the systems that you mentioned?
The scaling is absolutely trivial. That is multiplying the potential for the scaling factor. So it's the simplest thing you could imagine, mathematically. That's the scaling, yes, just moving it.
Yeah, but how do you know the scaling?
How do you know? This is very system dependent. Well, what I can say you is that a reasonable range is between 0.3 and 0.5 empirically. But you have to try to have an idea which are, because the very time that you get from your simulation is given by the combination of the restraints that you set and the scaling. So you cannot say, okay, I set this scaling, we'll get this time. No, it's not that trivial. You have to see what happens using the specific restraints. And the other question is that the second answer is you need at least 20 simulations. Statistically speaking, less than 20 you are in the small sample regimes. So statistically it's crazy to get to use less than 20 simulations. If you can afford 50, do it, obviously.
Okay, great. Well, let's thank all of our speakers again. Thank you. And I want to especially just acknowledge the 3 presenters here. Obviously, there's a lot more presentations coming, but in particular, just the idea of having software that's actually available to test. So there's all kinds of great questions. There's all kinds of great theory that one could ask, but all 3 of these, and the reason we chose these specific presentations was because these are pieces of software that could be tested right now on your systems. And they put a lot of effort not into just the theory and the underlying physics, but the kind of the whole pipeline to go from molecules to answers and to span that spectrum in between. So so that's the idea here, and again, there's a lot more presentations along these lines throughout the course of the next 4 days here. So as was said, people thought there were 4 presentations. There's 3. I'm not going to present. I'm going to just thank you all for coming. I will say that we did run out of booze earlier today, so I have to say I'm quite proud of our attendance here and all of your participation. Regarding extrapolation versus interpolation, I don't know what will happen if there's not any more alcohol in the back right there, so I'm hoping that we're still in an interpolation regime and we've been restocked in the back there. Tomorrow morning, just so everybody knows, up at Novartis, 8:30 coffee and registration and everything like that. 9 o'clock is kickoff, I believe, if I'm not incorrect. Somebody please correct me if I'm wrong here. Is there anything else that anybody from the organizing committee wants to say for the meeting, just to let everybody know what to be prepared for? Anybody from Novartis that needs to tell anybody about what happens when people show up? Are there any caveats, any security clearance things? Is anybody going to get patted down or anything like that? Or do we need to be careful? Is it obvious? It sounds like we can all get in all right here. Yeah, so it's right up on Mass Ave. It's a beautiful building off to the side. It's a big campus. You just kind of come into the side, and there'll be an entrance. It'll be pretty obvious right across from Flower Bakery. So if Novartis shows 10 different buildings all over, because they have a bunch of buildings all over, look for Flower Bakery up on Mass Ave, and the entrance is right next to there. And once you get in on there, and that's Flower in Cambridge, not in Boston. So with that, let's just thank all of our presenters and the organizers and everybody again, and thank you all for coming tonight. And it's been a great start to the meeting. And with that, let's resume the activities for the evening, and hopefully we're restocked, as I mentioned. And yeah, enjoy. And yeah, thanks again. Yeah, enjoy the conversations, discussions. Thanks to the presenters and the sponsors and everybody.